The goal of this lecture is that by the end of the lecture, you're able to define what adult stem cells are, define simple replication versus self-renewing, and name tissues that use these processes for regeneration. Describe the differences in regeneration capacity between the heart and the skeletal muscles. So the field of adult stem cell is relatively new. It was not until after 1950 that we started gathering data about the system of stem cells in different tissues. Before that, they thought is that we were born with all the organs. And as we age, there is damage to all the organs, and this cumulative damage to the organs results in aging and death. We know a lot more now, and we know that there's lots of different tissues that have active stem cells that help repair the tissue, repopulate all cells. But we still age and we still die. Most times we die because of failure to organs. So the regeneration capacity is not quite as well understood, and we would like to focus more research effort on that to help the regeneration capacity. So the first evidence that adult stem cells exist came from studies performed by Charles Lebon, a Canadian scientist. He lived to be 97, and he died a couple of years ago in 2007. And so Charles Lebon did some very clever studies using radioactive thymidine labeling in rats. And he was able to classify different organs based on the capacity of the cells to acquire thymidine. And he classified the organs in between high turnover organs. Those were self-renewing organs. Those are the blood or the bone marrow, skin, gut, and the epithelium of the gut and the skin. Low turnover organs, which could replicate but by a different process, which he calls simple duplication. And those are the liver, kidney, pancreas, vascular smooth muscle cells, and vascular endothelium. And those organs where there wasn't any uptake of the thymidine, and which he called static organs. And that was the central nervous system and the heart. So based on that definition of stem cells in different tissues, the criteria for stem cells is that stem cells can self-renew. And that means that in each cell division, they can either divide by asymmetric or symmetric division, but they have to give rise to at least one daughter cell that is identical. That a single stem cell is capable of differentiating into multiple functional cell types, and that stem cells can functionally reconstitute or regenerate a given tissue over a lifetime. So this is the definition of adult stem cells. And the first evidence of adult stem cells came from studies in hematopoietic stem cell transplantation done by Till and McCulloch, where they injected bone marrow into lethally irradiated mice, and they noticed that the levels of white blood cells decreased drastically after irradiation, but that the bone marrow cells that are transplanted can reconstitute all the blood cell types. And that was in 1963, and 5 years later, it was the first successful bone marrow transplantation in SCID people with severe combined immunodeficiency syndrome. And since the studies of adult stem cells has advanced tremendously, and the example is mainly on hematopoietic stem cells, where now we actually can build a hierarchy tree using cell surface markers to look at the progenitor cells from the very primitive stem cells down to the progenitor cells to the more differentiated cells that can give rise to all three lineages: erythroid, myeloid, and lymphocytes. How about the other tissues that Leblanc described as high turnover tissues? Let's talk about the epithelial stem cell in the intestine. It has been described that there is these stem cells depicted here in blue in the deep crypts of the intestine that can give rise to pretty much all the specialized cells of the intestine: assortative, goblet cells, Paneth cells, enteroendocrine cells. A similar kind of cell has been found in the skin, and actually two cell types has been found in the skin. One at the bulge of the hair follicle, and another one right at the basal layer of the epidermis, here depicted in blue. Whether they are related to the same origin is not quite clear yet, but they can give rise to hair and skin. And how about the skeletal muscle, which is one of the main organs that I study in my lab? So actually, the first evidence that skeletal muscle is a regenerative muscle and that it harbors a stem cell came from studies done by LeBlanc. So LeBlanc noticed that there is an age-related increase in the number of myonuclei. And using this thymidine labeling, he noticed that 24 hours after injection of the radioactive thymidine, there's these cells that are located beneath the basal lamina, but above the plasma membrane of myotube or myofibers, which he calls Saral cells. Here they're depicted because they express a specific transcription factor in red, PAX7. And these cells were labeled with thymidine, only a small percentage, 3%. But if he followed the cells 48 hours later, then he noticed that there were some labeled myonuclei inside the myofibers, and some remain labeled in the Saral cell position. So he made the assumption, which he was correct at the moment, that the Saral cells would divide and give rise to cells that would fuse with the myofibers, and that the Saral cells were the stem cells of the skeletal muscle. So this is the first evidence of stem cells in the skeletal muscle by studies done by LeBlanc in 1970. Well, how well can the skeletal muscle regenerate? And let me give you an example. I ran the Seattle Half Marathon last Sunday, and I'm not very athletic, so I was very sore after the half marathon, but I feel good now. And so I don't know if you watched The Biggest Loser last night, but they did a full marathon. This is people that weigh more than 200 pounds. So obviously they were also very sore after the full marathon. And there has been studies done back in the 70s where they have measured the level of creatine kinase after running a marathon, and the levels can go from 10 to 20 folds. So there's damage, and there's leakiness of the muscle in all the legs. But the muscle is capable of repairing all this damage, and in a couple of days, it can regenerate the damaged tissue. And I would consider that sort of a mild injury compared to the kind of injury we induce in mice in the laboratory, which we do so that we can understand better the regenerative process of the skeletal muscle. And so the injury that we do is that we use very high concentrated dose of cardiotoxin, which is snake venom from the cobra, injected directly in the tibialis anterior muscle of mice. And that causes massive necrosis. This is at day 3. You can see very necrotic tissue, very congested. The necrosis start being resolving by day 7, and then by day 14, there's full regeneration. So there is a tremendous robust capacity of the skeletal muscle to regenerate, and we're using this system to understand better what the stem cells are doing. So as you heard earlier from my grad student Nick Aronimakis, we have developed a flow cytometry assay, multicolor flow cytometry assay, to isolate and study the stem cells of the skeletal muscle, but also other kind of cell population from the skeletal muscle. And so the first thing we do is that we gate out all the hematopoietic cells. Here, we look only at the 45 negative cells. And we use markers that have been characterized for some kind of stem cells or other populations like endothelial cells. And if we look at what we would consider markers that are specific for the stem cells of the skeletal muscle, saddle cells, based on their definition of their anatomic position, they're beneath the basal lamina, and they express the receptor that binds laminin, which is alpha-7 integrin. We can isolate very pure population of saddle cells that are alpha-7 integrin positive. And if we look at the expression of CD34, which is a marker that has been used to characterize saddle cells as well as other kind of stem cells, we find two populations, 34 positive saddle cells and 34 negative saddle cells. These populations can be cultured as single cells in the dish, and they would twitch, and they seem identical to us in terms of the capacity to make colony of saddle cells, to differentiate. And if we look at all different muscle types in the mice, we see these populations that are present constantly in all the muscle. So we wonder, what's the role of these two different kind of saddle cells during injury? And we do similar studies to LeBlanc, where we injected nucleotide analog to follow the proliferation of these cells after injury. So without any injury, if we inject BRDU, which is a time-eating analog, for 3 days, and we harvest the muscle, digest it, isolate the saddle cells, and use an antibody specific for BRDU, fluorescently labeled, there's really not much uptake of BRDU in the saddle cell population. CD34 positive here depicted in blue, or CD34 negative in red, compared to, let's say, the bone marrow cells, which uptake very high level of BRDU in 3 days. However, after injury with cardiotoxin, here we inject the cardiotoxin, and at the same time we started giving BRDU. We gave BRDU at the peak of proliferation. There has been studies that have shown that the peak of proliferation after cardiotoxin injury in the muscle is from day 2.5 to day 3. We gave BRDU for 12 hours. We harvest the tissue and analyze it, and we noticed that the two populations, CD34 positive in blue and CD34 positive negative in red, were able to uptake very high level of BRDU. So they were actively proliferating, which is what is expected the stem cells to do after injury. If we then do a study where we give BRDU at the moment of high proliferation for the first 3 days after injury, and then we chase the fate of the cells until full regeneration, which is day 14. And then we harvest the animals and the muscle and analyze the two cells population by flow cytometry, what we notice is that CD34 positive cells are able to return BRDU. That means they were proliferative in the first 3 days, but they did not proliferate much after day 3 until day 14. But then the CD34 negative cells decrease the level of BRDU by 1 log compared to what they were at day 3, meaning that they had divided, and they probably divided for more than 3 divisions during that period of day 4 to day 14. If we then look at the muscle, what's going on in the muscle 3 days after injury, and then we stain for the markers that are specific for differentiation, myoblast fusion, one of them is myogenin, that would tell us which population is really contributing to muscle fusion and differentiation. And we see lots of myogenin positive cells here in red. Myogenin is a nuclear transcription factor, so it's in the nuclei. And if we stain for CD34, we see that the majority of the cells that are myogenin positive are CD34 negative. We can then go on and confirm this by sorting out these cells by flow cytometry, collecting the RNA, and performing quantitative RT-PCR for transcription factor specific for muscle regulatory factors, MI5, MyoD, just tells us that the cells are committed in the myogenic lineage. versus myogenic, which tells that the cells are really becoming differentiating and fusing with muscle fibers. What we notice is that CD34 negative cells express much higher levels of myogenic than the CD34 positive cells, which tells this is the majority population that's becoming differentiated and diffusing with muscle and is contributing to muscle regeneration. We can sort these cells and we can perform transplant directly into the muscle. And so in order to prove that these cells that we call CD34 negative are really myogenic, we did that. We sorted the cells, transplanted that in the muscle, direct injection into the muscle. And the cells were derived from transgenic mice. Transgenic mice that express nuclear laxity driven by one of these muscle regulatory specific factors, in this case MIF5, just so that we can prove that they're myogenic and we can track them in vivo by doing laxity staining. And here we see green new muscle fibers with stain for smooth muscle actin, which is a marker expressed by newly regenerated muscle fibers. And we can also see that they express beta-gal in their nuclei, which is depicted in red here. So the majority of the cells fuse and make new muscle fibers, of the majority of these CD34 negative cells. But also, some of them can revert back to the cytosol cell position, localize themselves to the cytosol cell position, and re-express CD34. And so the sample is given here. If you follow this cell that's beta-gal positive, actually this one here, and do a CD34 staining, we can prove that this cell, which was originally derived from CD34 negative cells before the transplant, after being transplanted in the muscle, can re-express CD34. So the working hypothesis, we're still working this out, is that there's two main populations of cytosol. One that express CD34. We believe these are the quiescent cells because they express very high level of the myogenic quiescent markers. Upon activation after injury, many of these cells can proliferate since we see a peak of BRDU uptake for both CD34 positive and CD34 negative cells. The majority of the CD34 negative cells keep on proliferating during the process of regeneration of the skeletal muscle and start expressing high level of myogenic and differentiate and form new myofibers. And some of them can actually revert back to be cytosol cells, quiescent cytosol cells, because they can re-express CD34. So this is a working hypothesis, but it fits with all the results in injury. So I have described you samples of highly regenerative tissue. We talk about the bone marrow, we talk about the skin, we talk about the GI. How about other tissues that Leblanc characterized as not really high turnover tissue, but low turnover tissue, and he thought that they were regenerated by single duplication. One of them is the liver. However, the liver does have tremendous regeneration capacity, and it is known back in ancient Greece mythology. We know that based on the Prometheus myth, you guys remember the Prometheus myth? So Prometheus gave the power of fire to mortals, and he was punished by Zeus, and he was chained in the tip of a mountain on a rock, I believe. And the vulture would come in every evening and eat the liver. By morning, the liver had completely regenerated. Obviously, this is an exaggeration, but it is known that the liver has tremendous regeneration capacity. Studies done by Canales in the 19th century show that you could do partial liver resection. Up to 2/3 of the liver can be removed, and in 5 to 7 days, it can be regenerated. So how does this happen? This really has not been in stem cell discovered in the liver yet, but what is thought to be happening in the liver is that mature hepatocytes, after injury, can proliferate and make more of their own. And this is called simple duplication. And what's amazing about it is that you know how the liver has different functions. So there's areas of hepatocytes, areas of the liver of hepatocytes that are specialized in detoxification. So they have very high level of P450. And there are other areas of the liver that have more production capacity of like albumin. Well, they can maintain their function as they proliferate and make more of their own. So it's a very interesting process of regeneration. How about other tissues? Let's say the pancreas. So Douglas Melton, who is the co-director of Harvard Stem Cell Institute, a very, very smart developmental biology scientist, has dedicated his research focus on studying regeneration of the pancreas because he has 2 kids that have diabetes type 2. And so he engineered these transgenic mice. Nowadays you can make transgenic mice that would express certain reporter genes driven by specific tissue promoters. So he engineered a mouse that has tamoxifen-inducible insulin promoter CRE. And CRE is a DNA recombinase protein that's capable of cleaving and removing DNA fragments and joining these DNA fragments. And so he engineered a mouse that has a transgene inducible tamoxifen. So only when he gives tamoxifen, the cells that are expressing insulin, only the cells that have insulin promoter would express CRE. And then CRE would recognize another transgene in the same genome of the mouse that would express alkaline phosphatase, but it's silent because it has a stop codon flanked by the flanking region of the CRE recombinase. So when CRE is activated, it would excite the stop codon, and now this reporter gene, in this case alkaline phosphatase, gets to be expressed only in the cells that are expressing insulin. So everybody follow me? Because I'm going to give another sample like this kind of inducible system. So what he did is he gave tamoxifen to these mice. And so all the beta islet producing cells, all the insulin producing cells would turn alkaline phosphatase positive, and then you could give them a chromogen and they would turn blue. And so he followed these mice after he had converted all the insulin producing cells into alkaline phosphatase positive. He followed them with time and injury. And what he found is that, let's say, if indeed this organ is replicating or reproducing by simple duplication, so the cells that are already alkaline phosphatase after injury, they will proliferate and they give rise to more alkaline phosphatase positive cells. And this is a irreversible recombination. So once they turn alkaline phosphatase positive, they will be alkaline phosphatase positive forever, and all their daughters too. So if indeed the new cells come from insulin-producing cells, the newer cells will be alkaline phosphatase positive after injury. But if there is a stem cell that was not insulin producing at the moment he was giving tamoxifen, and those are the stem cells that are giving rise to new cells that are regenerating the tissue, then the percentage of alkaline phosphatase positive cells or blue cells will decrease with time and injury, and that will be a self-renewing system. So what Douglas Melton found is that the pancreas can indeed regenerate after injury, but this happens by simple duplication because the number of alkaline phosphatase positive cells after injury remain the same. So, and how about the heart? Is that the case for the heart? Well, LeBlanc showed that the heart is one of these organs that had no uptake of thymidine. So he called it static organ. And it is the consensus up till recently that the heart doesn't really regenerate, doesn't have a stem cell capacity, because we die of heart attack, and heart disease kills more people than any other condition. And the way it happens is that we have atherosclerotic plaques in coronary arteries that supply blood to the heart. When they get blocked, then there is ischemia to the area of the heart, and because there's no regeneration, that area of the heart dies and is replaced by fibrotic tissue. So a similar approach was used to understand what happens to the heart after injury. A tamoxifen-inducible system that expresses Cre under a tissue-specific promoter, here under cardiac myosin heavy chain promoter. So only cells that are mature cardiomyocytes will be able to express Cre when tamoxifen is given. And there's also a reporter gene. The cells are blue originally, but if Cre gets activated, then LAC-C gets turned off, but now GFP gets turned on. So before the tamoxifen is given, all the cardiomyocytes are LAC-C positive, they're all blue. After tamoxifen is given, all the cardiomyocytes become GFP positive. If there's injury and if the heart does have a stem cell regeneration capacity, that cells were not cardiomyocyte positive, they were not expressing cardiomyosin heavy chain at the moment tamoxifen is given. After an insult to the heart, then the new regenerating cardiomyocytes will have been derived from cells that are not GFP positive, that are LAC-C positive. Whereas if there's just simple duplication, then the heart will remain the majority of the cardiomyocyte GFP positive. Is everybody follow me here? Okay, so he did that experiment, and with time, really not much happened. The majority of the cardiomyocytes were green, were GFP positive. After myocardial infarction, he noticed that in the area of the injury, there were lots of new regenerating cardiomyocytes that were not GFP positive, that were LAC-C positive. So it does seem that the heart has a stem cell population that's capable of renewing after injury. So this is the first evidence that there could be a stem cell in the heart. And here is the results. Majority of the cells before injury are GFP positive. After myocardial infarction, there's some replacement of cardiomyocyte by cells that are not GFP positive anymore, but they're beta-gal positive. Here they did the staining for beta-gal in red. Okay. But even more recently, and I would consider this the strongest evidence of cardiomyocyte renewal, is a very clever study that was done just recently, in which they took advantage of the high level of carbon-14 in the atmosphere after the Cold War. And so what they noticed is that plants would incorporate carbon-14 via photosynthesis. We incorporate carbon-14 because we eat plants and we eat animals that eat plants. And so if you follow the level of atmospheric carbon-14 after the Cold War, you could plot a graph. Very high after 1960, and it decreases. So you could sort of tell the level of carbon-14 if you know the age of a person. So for here, they drew a vertical line for a person that was born in 1970. So at the moment that person was born, all the cells at the moment that were born had this level of carbon-14 right here. If this person had organs that had never regenerated, they will have the same age as the age of the person. So if you measure the level of carbon-14 in the organs of this patient that had never regenerated, they will have the same level of carbon-14, and they will be plotted right here, where the vertical line meets the graph of the level of carbon-14. But if you have organs that have regenerated, that have new cells that were born after you were born, then the level of carbon-14 will be lower. And so that's exactly what they found. They looked at the heart of patients that died and patients that were born after 1960, and they noticed that the heart cells always were younger than the age of the patients. And they went on and analyzed specifically cardiomyocytes from the heart of these patients. They sorted out cardiomyocytes that were troponin T positive, look at the level of carbon-14, and they noticed that the level of carbon-14 were always lower than the age of the patient. And using a mathematical algorithm, they found that in young patients, there is 1% turnover of cardiomyocytes per year, and that decreases to 0.45% in older people older than 75 years old. So they calculated that through our lifespan, we can replace up to 50% of our cardiomyocytes, which is fairly significant. Okay, so how about some other evidence of cardiomyocyte renewal? In my lab, we work with muscular dystrophy, and we have mouse model to study skeletal muscle regeneration to treat muscular dystrophy. And there's one way that we could track the number of muscle fibers that are damaged, and that's using Evans blue. It's a dye has been used for more than 80 years to track plasma volume because it binds albumin and it diffuses with albumin. If the muscle fibers are damaged, they would trap this blue, and they would turn blue, the muscle fibers. And so it's well known that the muscles in mice with muscular dystrophy will trap this dye because they're damaged. What we didn't know, which we just made an observation recently, is that there are also cardiomyocytes in the heart of these mice with muscular dystrophy that can retain the dye. And so let's take the diaphragm, which is one of the muscles that's mostly severely affected with muscular dystrophy. You see the number of muscle fibers that uptake the dye. This dye is blue, but can emit fluorescence in the spectrum of red and far red. We see lots of muscle fibers that can retain the dye. And in the heart, we have areas of cardiomyocytes that are damaged. So that tells us that there is damage of the heart in these mice. And there has been a well-described dilated cardiomyopathy in patients with Duchenne muscular dystrophy, but the pathogenesis of the dilated cardiomyopathy has not really been well understood. And here is an area of cardiomyocytes that are damaged. And that just tells us that the dystrophy is somehow affecting the cardiomyocytes. What we didn't know is if these cardiomyocytes are replaced by fibrotic tissue, which is normally what happens during myocardial infarction, or is they actually replaced by new cardiomyocytes that can regenerate the damaged tissue. So we stain with a proliferation marker, Ki67, which is a nuclear protein expressed only by dividing cells. What we noticed is that there were proliferating cells near the damaged cardiomyocytes. And if we do BRDU labeling, we also find that there are BRDU positive cardiomyocytes that stain troponin T positive in these hearts, whereas in wild-type hearts from normal mice, there's no BRDU labeling. And so this is one of these cases where there is a troponin positive cardiomyocyte that is BRDU positive. So that tells us that there are proliferating cardiomyocytes in the heart of mice with muscular dystrophy, but we still don't know if they're produced by simple duplication of cell renewing. So that's something that's research that's going on in my lab right now. So we started looking at potential cardiac stem cells, and I'm just going to call them putative cardiac stem cells because the literature is very controversial. And there has been some markers that have been used to characterize cardiac stem cells, and some of the ones that people have described are the stem cell marker SCA1, which we use in our antibody cocktail when we look at flow cytometry for the muscle, and we decided to study these cells. Once more, we exclude the hematopoietic cells. We only look at the 45 negative cells. And there's two populations that are SCA1 positive, the endothelial cells, which we have well characterized in the skeletal muscle that express SCA1 and CD31. And there's this perivascular population that express SCA1 but not CD31, so it's not really endothelial specific, but it's located near vessels. And that population also expresses CD34. And so we looked at the distribution of this population in the atrium and the ventricle. What we noticed is there's much higher level of SCA1 positive cells in the atrium. And by the way, SCA1 is just one of these markers that people have reported to be expressed by cardiac progenitor cells. So that's why we're looking at this population. We can culture these cells in stem cell media, and only the cells from the atrium can be expanded in vitro, but the cells from the ventricle cannot grow very well. And the cells that have been kept in the expansion media would express some of the transcription factors that are specific for cardiac progenitor cells. NKX2.5 is one of these transcription factors that is specific for cardiac progenitor cells. Is important for cardiac differentiation. Undifferentiated cells are negative for some of the mature contractile proteins for cardiomyocytes, so they're negative for cardiac troponin. But after differentiation with oxytocin, and once more we're following the protocols that has been used in the literature, they start expressing some of these cardiac specific proteins. This is MF20, is an antibody for sarcomeric actin, so they also express some of these cardiac contractile proteins. And this is the corroboration by RT-PCR that they express these transcription factors. These are undifferentiated cells in lane 1 are relatively negative. After differentiation with oxytocin, they express some of these transcription factors like GATA4, NKX2.5 is specific for cardiogenesis, and also mature contractile proteins like cardiac troponin I. So we decided to see what happens to these cells in vivo. And since we use the skeletal muscle of one as our bread and butter studies for transplantation, we decided to transplant the cells into the skeletal muscle, just like how we transplant adult cells, and see what they do in the skeletal muscle environment. And the cells were derived from transgenic mice that expressed green fluorescent protein. They were cultured in vitro and transplanted directly into the tibialis anterior muscle. And here they are, the cells that are GFP positive. They seem to be making muscle fibers, but when we stain them for all the skeletal myosin antibodies, they were not positive for skeletal myosins, but they were positive for cardiac troponin T, which is specific for cardiac muscle, and it was not expressed in skeletal muscle at all. So they seem to be making what seems to us like cardiac-like cells in the skeletal muscle. Here is the injection tract, and here is some cells in magenta expressing cardiac troponin T. And they also express muscle actin, which is a marker expressed by embryonic stem cells as they differentiate into cardiomyocytes. And so here is a longitudinal section of the skeletal muscle cells that were transplanted were GFP positive. Here is cardiac troponin T. And they seem to be making the right kind of striation that cardiac muscle will make, but it's not present in the myotube or the myofibers of the skeletal muscle. So they seem to be making what we believe is cardiomyocyte in the skeletal muscle. So we decided to transplant the cells in the heart, same delivery system, directly in the ventricle of the heart. And because the heart is very autofluorescent, especially for the green fluorescent spectrum, we also label the cells with PKH26, which is a red fluorescent dye, to track the cells in the heart muscle. So here are the cells 2 weeks after transplant. But when we stained with troponin, they were not troponin positive, and indeed the Asian E was very disappointing because it looked like very fibrotic tissue. And when we stained with trichome, which stains collagen in blue, right at the injection site, we see lots of fibrosis in the heart. So why can't the heart regenerate like the skeletal muscle? We don't really know, but the findings of our studies in the lab indicate that perhaps the skeletal muscle is more permissive than the heart. And so we're very interested in studying what are the growth factors, what is the interaction with the other nursing cells in the environment, what's the role of the inflammation that would allow the stem cells to make cardiomyocytes in the skeletal muscle, but not in the heart. So the idea that the heart doesn't have a stem cell has pretty much been ruled out. There seems to be a stem cell population in the heart. It's just not very efficient at making new cardiomyocytes. Some people have claimed that the number of stem cells in the heart are not sufficient. That could be true compared to other organs like the skeletal muscle, where there's 2 to 5% of cytal cells. But you don't really need enough number of stem cells if they're really doing a good job of regenerating. So I don't quite buy that idea. Some people claim that the severity of injury, myocardial infarction, is just too severe, which in a way I agree, but the results I show you with the cardiotoxin injury, where we basically kill the whole muscle and we get full regeneration in 2 weeks in the skeletal muscle, which tells us that that's a very severe injury model, and we get full regeneration. So the severity is not quite the answer. How about acuteness of the injury? That could be, because when we injure these mice with cardiotoxin, they're limping for 1 week. They're not using that leg, so they're allowing that leg to repair. We don't have the luxury to put away our heart and let it regenerate and then put it back into us. The heart needs to be beating constantly. So perhaps the heart is compensating by a rapid repair mechanism, which is not necessarily the most efficient, which normally results in a scar formation or fibrosis. And so the tools that we have in the lab, models of muscular dystrophy, where we do see some degree of cardiac regeneration that doesn't result in fibrosis, would be helpful to understand better how to improve regeneration of the heart, and also the environment. Why do the cells can sort of make cardiomyocytes in the skeletal muscle but not in the heart? That's very intriguing, and so we're looking at that, the environmental factor, the nursing cells, the niche, and also the inflammatory component. So I remember being here 6 years ago when I was first year resident in clinical pathology, and I finished with this, and I decided to still finish with this. I remember giving a talk about my work with muscular dystrophy and how we're looking for therapy for muscular dystrophy, and how at the moment when my daughter was 3 years old, asked me what kind of work I do, and I said I'm working with skeletal muscle to make skeletal muscle stronger, and her solution was eat spinach and make muscle like Popeye. I didn't have a good answer for her at that moment. I still don't have a good answer for her 6 years later. This is her in Halloween. So she still asks me what I do at work. And my answer is I'm still looking for the magic spinach to make muscle stronger. I do have a little bit of justification because now I'm looking at the heart too. It's a bit more complicated. It's not just a simple spinach, and this magic spinach may also help stronger hearts. So let me acknowledge the people in my lab that have done the work. And I don't have a picture of an undergrad student that have done some of the work in the heart of muscular dystrophy mice, Aislinn Hayes. Nick Aronimakis, a grad student in pathology, you heard him talking today about the skeletal muscle regeneration. Gayatri has been a technician in my lab and just left recently. She did some of the molecular biology work that I described today. And Kanit is a fellow that came from Thailand and has just left and returned back to Thailand. And he did some of the work that I showed with the cardiac progenitor cells. And I want to acknowledge my departments, pathology and lab medicine, NIANS and NIH for the funding, and the GEM Foundation for the funding. Thank you.
Morayma, can you explain why in the heart in your muscular dystrophy model there is damage?
It's not well understood. There is dilated cardiomyopathy, and the thought is that because the muscles in our bodies in patients with Duchenne muscular dystrophy are so weak, there is a compensatory mechanism of the heart to work harder because of the overload and become dilated. The fact that there is foci of cardiomyocytes that are damaged suggests that there is microischemia to those cardiomyocytes. But it's not really well understood, and that's very interesting. Also, the observation is that it seems to be a chronic but minor injury, and there seems to be better repair in those circumstances that when you get myocardial infarction, where you got massive damage and ischemia to a big area of the heart, and the solution is, or the compensation of the body is fibrosis. Whereas in this model, we don't see much fibrosis in the heart, and there seems to be a replacement of the cardiomyocyte. So it perhaps is because it's less acute and more chronic, that there could be some sort of a stem cell that can regenerate those cardiomyocytes. But we're still working hard on trying to understand the pathogenesis of why there's microischemia in the heart. The thought that the cardiomyocytes may need dystrophin, and if they don't have dystrophin in every contraction, they may have muscle damage, has also been posed. Dr. Chandler?
I mean, the other thing I guess that makes the heart a little bit different is the myocytes all have to contract in unison, or at least in the right rhythm, and that the electrical activity in the heart, the stimulation activity is much more tightly regulated than it is in muscle, and that maybe you could regenerate a few myocytes in the heart, but if you had to regenerate a large area, if it wasn't conducting, and then do you have the risk of fibrillation or arrhythmia or something else, and that's why you can't regenerate? I mean, is there some other thing that it's blocked in the heart because it just doesn't work for large numbers growing up?
That is a great point, and indeed, one of the biggest hope for cardiac regeneration was in clinical trials that were conducted over 10 years ago in Europe, where they were doing transplant of skeletal myoblast because they can't make a skeletal muscle in the heart, and to some extent they can synchronize with cardiomyocytes. But what they found in patients that they followed for a long time in these clinical trials is that many patients would have arrhythmias because the skeletal muscle, that even though was grafted, was not contracting at the same pace as the cardiomyocytes.
So along that same line, I would just ask, has anyone taken the cardiotoxin system, degenerated, caused necrosis in a femur or in a tibialis muscle or something like that, and then looked at the regeneration while tonically contracting it once a second for 2 weeks? So to sort of maintain, so make it heart-like, and so it actually is under the same stress, metabolic demand, and all that stuff. And can it still actually regenerate if it would have to do what a heart would have to do? Because heart, you can't really limp on your heart, but you could conceivably on your leg.
That's a very good idea. I don't know of anybody that have done that. What I've known is that, like in the cardiotoxin injury model in rats, just to prove that the skeletal muscles have a tremendous regeneration capacity, is that they can give cardiotoxin once every week for 60 weeks in a row, and the skeletal muscle will regenerate. But I don't think anybody had really followed what happens with the heart if you do that. So that's a good point.
I was wondering what happens with ventricular hypertrophy and whether the growth that is seen in heart failure and so on is, if you think there's a role for stem cells in that muscular hypertrophy, or is there other mechanisms? And I guess the other side of the coin is sometimes people are put on left ventricular assistive devices for weeks and months at a time, and sometimes the heart seems to do better after you take them off the LVAD. Is there a role for stem cells in those kinds of
That's a
situations?
good point. And like I say, it wasn't until just recently, with those two papers, one in 2007, one just recently in Science in 2009, that it has been accepted that there are possibly stem cells that can regenerate the heart, although not very efficiently. So I don't think anybody have looked at any of this before. And the idea that in left ventricular hypertrophy, what just happens is that the cardiomyocytes get bigger. That was the whole idea in the past, but whether there's contribution of a stem cell population, I don't think people have really looked at. The best way to look at it is by doing this kind of nucleotide analog tracing, but they cannot do that in humans. There has been some studies where they have looked at sex mismatch bone marrow transplantation, and they have found some contribution of bone marrow cells, but it's very minor.
I was just thinking, if you took the heart transplant patients here and did those studies that were done with nuclear, environmental carbon-14 exposure, we actually have all those hearts that are obtained. Some of them have been on LVADs, kind of will have been through all this.
I would, I would just contact the German authors and ask them to collaborate, because if you have a good source to start looking at all these questions, then it's worth it. But yeah, I don't think anybody have looked at any of these for heart regeneration. Like I said, there has been some studies that have done sex mismatch transplant. There has been also studies, not just bone marrow transplant of heart, and they have seen some minor contribution. It may correlate to the 1% turnover that was shown in this carbon-14 study, which is not enough to really repair the heart after ischemia attack.
I have one more question myself, unless, was there a question in the audience? How about embryonic stem cells? Can those be made to differentiate into cardiac muscle?
Oh yes, and indeed it seems to be one of the easiest default pathway for embryonic stem cells. And I understand you heard Chuck Murray talking here last year, and that is the focus of his research, is to use embryonic stem cells. Now he's more involved into IPF, induced pluripotent cells, and their differentiation into cardiomyocytes. And yes, it is an avenue. There has been pros and cons of every approach, but I would say that among all the different stem cell sources, that would be one that's very promising.
Thank you, Dr. Reyes.
Thank you.
Thank you.